In this video, we're going to talk a little bit more about the physical structure of DNA, how the different chemical molecules are oriented relative to one another, and how all that interaction works to kind of come together to form the DNA double helix. Okay, so DNA, as I've said in previous videos, forms a double helix. All right, and the reason that that helix can form is based on the sugar that's associated with the nucleotide associated with DNA. All right, so at that 2' carbon, we've got a hydrogen. 3' carbon is a hydroxyl group. 5' carbon, all right, is where our phosphate's attached. So let me fill those in. There's 3' there, 2', 1', okay? First carbon is where our nitrogenous base is connected. Remember, what makes up our nucleotide is the phosphate, the sugar, and the nitrogenous base, okay? Now, when DNA replicates, it's going to add the new nucleotide to the 3' end. So what's going to happen is our phosphate group from our next nucleotide is going to form a bond at that 3' carbon, all right? That's going to give us, and I'm using the two different colors just so you can see the difference between the first nucleotide and the second nucleotide, okay? So that's going to give us now our next nucleotide that's added on, okay? And then we'll have another 3' hydroxyl group here. We'll have the hydrogen here, okay? Where that hydrogen comes into play, because hydrogen is very small, it doesn't create any physical encumbrances, or it doesn't interfere with the bases pairing in the middle of our DNA double helix, okay? If this was a hydroxyl group, that hydroxyl group is bulky enough, it prevents the bases from being able to connect to one another to make hydrogen bonds, okay? So having the hydrogen at that 2' carbon is what makes this a DNA molecule, and that's important in establishing the double helix, okay? So I drew a bond between our first and second nucleotide. This bond is a covalent bond, but it's a covalent bond that has a very specific name. It's called a phosphodiester bond, okay? So that phosphodiester bond is going to connect our two nucleotides together, all right? And just so we have it on this side too, all right? I didn't write base upside down because I'm not quite that talented. Plus, it'd be harder for you to read, so really it was all for your benefit, okay? Ooh, squeakiness. We've got a phosphate here. We'd have our hydroxyl group here, our hydrogen here, because this is going to be our 1', 2', 3', 4', 5' carbon, okay? And we're going to make a bond between the nucleotide here and here, okay? Again, this would be a phosphodiester bond. And our phosphodiester bond is a type of covalent bond, okay? But if you notice what happens with the way I drew this, okay? We've got our 5' carbon up here. So that's what we label as the 5' end of our DNA molecule. We've got a free 3' end down here. So that's going to be our 3' end, okay? And that DNA molecule is going to run from 5' to 3'. That's how new bases are added to our DNA double helix during DNA replication. Always have to add the new nucleotide to the 3' end. I know I drew it over here and it looked like I added it to the 5' end. All right, that's just so that I could fill in this drawing. In reality, on this molecule over here, this is where a new nucleotide would be added. Okay, just like down here, we can add a new nucleotide here. Okay, it has to be added because what we've got here, we've got 3', 1, 2, this is going to be 5'. Okay, so on this DNA molecule, our 3' end is up here, our 5' end is here. All right, and oops, I totally drew that the wrong way, but fortunately I can fix that, right? So again, we're going to run 5' to 3'. Okay? So if you notice, these two are opposite one another. Okay? What that means is that DNA double helix is anti-parallel, meaning one strand runs one direction, the other strand runs the other direction. Okay? And what we're going to have happen in the center of our DNA molecule, this is where we're going to get our base pairing to happen. Okay? And those bases, remember, we have to have one purine, one pyrimidine. Okay? So our purine might be our adenine. All right? Our pyrimidine would be our thymine, in which case we'd have 2 hydrogen bonds holding that particular base pair together. Okay? It might be on this side, we might have our pyrimidine over here and our purine over here. Okay? So in this case, our purine would be guanine, our pyrimidine would be cytosine. We'd have 3 hydrogen bonds holding that together, okay? But what ends up happening, you've got to have a purine paired with a pyrimidine because remember, your purine has 2 rings, your pyrimidine has 1 ring in it. So that we maintain the space between our sugar phosphate backbone, you've got to have 3 rings in the center to maintain that distance. So we've got 1 ring here, 2 rings here. Okay? So that base pairing is going to be important in forming the steps of our DNA double helix. All right? So when DNA replication happens, and DNA replication is going to be our next topic, okay? You are going to have 1 strand of DNA that is going to be the template strand. The template strand is what I will also call the parental strand, okay? And you may, for example, have a particular sequence, all right? And I'm just randomly putting up letters here, okay? This may be our template strand, our parental strand, okay? It's going to run 5' to 3'. The complementary DNA strand, all right, of the parental DNA would be opposite, all right? If you have a G, you would put in C. If you have a T, you'd put in A. C, G, C, G, G goes with C, T goes with A goes with T. Okay? Now this strand is going to run the opposite direction. Okay? So either of these can function as the template strand. Okay? What happens during DNA replication? we get those hydrogen bonds to separate in the center. And what that does, okay, is it opens things up so that enzymes can come in and fill in our complementary strand. So this end would be 3', this end would be 5'. And what we do is our other strand, when it ran 3' to 5' here, right, was T, A, C, G, G, A, squeaky A, C, okay? Those enzymes are going to fill in the complementary strand to this one, which would be ATG, CC, TTG, all right? So in this case, what I've done, the blue is our new strand of DNA, our new synthesis, okay? The pink strand, this is our parental, or if you want to think of this as our original strand of DNA, okay? So you can, if you're given one set of sequences, you can fill in the other knowing the sequence of one, okay? Because remember, in terms of base pairing, A pairs with T, C pairs with G. Okay, we've got 3 hydrogen bonds between cytosine and guanine, 3 hydrogen bonds between adenine and thymine. All right, hopefully this makes sense. If you have any questions, please let me know.